We already discussed about the systemic acquired resistance in the last talk, and we know that the systemic acquired resistance is quite common and it is shown by all the plants in the field. We also talk that the basic requirements of the systemic acquired resistance is the production of hypersensitive response in the cells where a virulent pathogen first tried to attack the plant. And the second thing is that once the plant detect the A virulent pathogen, it not only show HAR or the hypersensitive response, but also it produce the salicylic acid. And that salicylic acid has a key role in the systemic acquired resistance because those mutants which unable to produce the salicylic acid also unable to produce the systemic acquired resistance. But if you recall, we also discussed that the salicylic acid, despite a key molecule in the systemic acquired resistance, but still it is not a signaling molecule in the systemic acquired resistance. So the question always pending before the plant pathologist that which gene actually regulate the systemic acquired resistance and which gene or the genes are responsible for the execution of the systemic acquired resistance who are also connected with the salicylic acid. The other pending question for the plant scientist is that why plant remember that it is once infected by the A virulent pathogen because plants unlike the animal don't have the T cell or the B cells, and similarly plant don't have antigen antibody interaction. So in the next slides, we try to briefly cover all these questions and get a complete picture about the systemic acquired resistance. We already discussed about the systemic acquired resistance, and we know that systemic acquired resistance is not possible without HAR in the local tissue, which later transform in the whole plant, and the plant show resistance against the virulent pathogen. So if the salicylic acid is not the signaling molecule, then what are the uh, other candidate which act as a signaling molecule for the systemic acquired resistance? In last few years, the scientists claim that there are other molecules which can act as as a signals of the systemic acquired resistance, which include methyl salicylic acid, jasmonic acid, azelaic acid, and many more. But still, we don't say with confidence that this molecule is definitely a signaling molecule, and it is still debatable in plant pathology that which molecule actually act as a signaling molecule in the systemic acquired resistance. Despite many papers are published and many candidates are reported. This slide is actually the extension of the previous slide or the first slide. And we already know that the salicylic acid is not the signaling molecule, but without the salicylic acid, SAR is not possible. And this is supported by the experiments which are done on the loss of function mutant SID2, and in this mutant the salicylic acid is not produced, and that mutant also not show any kind of systemic acquired resistance. And similarly the plants which are transformed with the Naji gene, where the salicylic acid is degraded, and these transformants are also not show the systemic acquired resistance. So this thing established that without the salicylic acid SAR not possible, but still salicylic acid is not the signaling molecule. The second thing, when people applied the salicylic acid on the plants, they noticed that the expression of a gene which is known as NPR1 gene is changed. And this so automatically the it means that this gene has some kind of role in the systemic acquired resistance, and now it is considered as a master regulator in the systemic acquired resistance. NPR1 gene is considered as a master regulator in the systemic acquired resistance, and it is also supported by the evidence that a loss of function of NPR1 gene also unable to show the SAR. It means without NPR1, the SAR not possible. The last is that the PR genes actually responsible for the execution of the systemic acquired resistance, and how the salicylic acid, NPR1 and PR1 genes are interrelated with each other. We are going to discuss in the next slide, and as a result of their interaction, the SAR is possible. This is very important part of today's talk because this slide tell us that how salicylic acid and the master regulator gene NPR1, which stand for non-expressor of PR gene, and PR genes, which stands for pathogenesis related protein, can produce the systemic acquired resistance. Here, there is a plant cell. Salicylic acid is produced in the chloroplast by a pathway which is known as ICS1 pathway. Most of the salicylic acid is produced, about 90% of the salicylic acid is produced by this pathway. And you also notice that there is the presence of NPR1, and this NPR1 gene is present in the cytoplasm, not in the nucleus. And this NPR1 gene is here in the form of oligomer. And when the plant is not under the attack of pathogen, it means the NPR1 gene is present in the inactive form. But once the plant is attacked by the avirulent pathogen, the amount of salicylic acid increased, not in the cell which show the HR, but all other cells present in the plant. And this increased level of salicylic acid change the redox state of the cell, and as a result of this change of redox state, the NPR1, which is present in inactive oligomer form, converted into the monomer form, and it is translocated into the nucleus. And once it is translocated into the nucleus, it interact with the PR genes by interacting with the transcription factor. And as a result of that, these PR genes are activated. These PR genes are basically uh, are the known for the production of the antimicrobial compounds such as glucanases or chitinases, and as a result of that, the plant pathogen which is virulent but it is killed by the plant. So the NPR1 is actually the master regulator, or it is a link between the salicylic acid and the PR genes, which are the executioner of the systemic acquired resistance. PR genes are the pathogenesis related genes, but some people say that this is not appropriate name for the PR genes because they are more related with resistance than the pathogenesis, And without the PR genes, the systemic acquired resistance is not possible. So PR genes are small proteins which on activation produce the antimicrobial compounds. So the question might be arises that if the PR genes are not activated, then the SAR is possible. No, SAR is not possible if the PR genes are not activated. So without HR, salicylic acid, NPR1 and PR genes, systemic acquired resistance is not possible. So, scientists discovered that the systemic acquired resistance associated immune memory remain in the plants not for the weeks, even for the months, or even for generations. And it is also fact that the plant cells don't have any kind of memory cells which are known as P or the T cells. So how it is possible that the plant remember that once it is infected by a pathogen? And this process is known as priming. Priming is little bit different from the vaccination, because in case of the priming, the plant tissues are sensitized against the pathogen, and this sensitization help them to counter the pathogen when it try to attack them. In their life, the plants are attacked by a variety of pathogens, and these pathogens vary when it comes to the mode of nutrition. Some pathogens are known as the biotrophs. Biotrophs who live in the living tissue. For example, all the plant viruses are biotroph. Similarly, those fungi which causes the rust diseases are also biotroph. Then comes the necrotroph. Necrotrophs are those pathogens which kill the host tissue and then live in the dead tissue. For example, the Alternaria species or the Botrytis species. Similarly, in between the biotroph and the necrotroph, there are the hemibiotrophs. Hemibiotrophs are those plant pathogens which first live as a biotroph, mean they live in the living tissue, and later they kill the tissue and start live as a necrotroph, mean in the dead tissue. Pseudomonas syringae is example of the hemibiotrophs. So biotrophs and the hemibiotrophs initially are the same one. As I mentioned that in its life, the plant is not only attacked by the biotrophs or the hemibiotroph or by the necrotroph. So what the happen in case of the plants, according to which pathogen it is under the attack, it activate the defense system. If the plant is under the attack of the biotroph or the hemibiotroph, then it produces a signaling molecule we already discussed. It is known as salicylic acid. So this salicylic acid production change the cell state and result in the activation of a gene which is known as NPR1 gene, which is also known as a master regulator. And this master regulator then activate another group of genes which are known as a PR genes, and plant become resistant, which is known as systemic acquired resistance. But if the plant is under the attack of the necrotroph, then it will not produce the salicylic acid, but it produce the jasmonic acid and the ethylene. So the molecule which act as a link is not the salicylic acid, but it is the jasmonic acid or the ethylene. And again, the signal is went to the NPR1, and the master regulator is again the NPR1. But again, there is a difference. Not this time the PR genes are activated, but another group of genes which are known as PDF 1.2 or Thai 1.2, these genes are activated and the plant become resistant. So you notice if the plant is attacked by the biotroph or the hemibiotroph, then it follow this pathway. But if the same plant is under the attack of necrotroph, then it follow this pathway. So how these two are interlinked? Scientists find out that the relationship between the salicylic acid and the jasmonic acid or ethylene is the antagonistic one. Means that when there is more production of salicylic acid, then it stops the production of jasmonic acid or the ethylene. It also means when the plant is under the attack of biotroph or hemibiotroph, it activate another pathway to defend itself by stopping the signaling molecule which is involved in the defense against the necrotroph. If similarly the plant is under the attack of necrotroph, then it try to stop the signaling pathway which has role in the defense against the biotroph or the hemibiotroph. So in short, it also means that plant at a one point not defend itself against both biotroph and the necrotroph. So I hope that everyone has a basic idea how the plants give a priority or activate the defense system according to the pathogen they are under the attack. Non-pathogens are very smart individuals, and because they are smart, that is why they are able to cause the disease in the plants, which are much bigger and much stronger. There is a one pathogen which is known as Pseudomonas syringae pathovar tomato. This pathogen is a hemibiotroph, and we already discussed that the plant defend against biotroph and hemibiotroph by the production of systemic acquired resistance. And the systemic acquired resistance is not possible without the production of salicylic acid. And we all know that the salicylic acid relationship with the jasmonic acid is a antagonistic one. Means if more is the production of the jasmonic acid, then less is the production of salicylic acid. What this pathogen actually do, it not produce the jasmonic acid but produce a mimic of jasmonic acid which is known as coronantine. It mimic means that it look like jasmonic acid, but it is not actually the jasmonic acid. And when this hormone is produced by the pathogen, the plant stop producing the salicylic acid. As we already know, the interaction between the or relationship between the jasmonic acid and the salicylic acid is the antagonistic one. So when the plant stop producing the salicylic acid, it means the SAR is not activated. And when the SAR is not activated, this a plant is able to cause the disease. So you can acknowledge the cleverness of the plant pathogen Pseudomonas syringae, how it confused the plant, as a result of that, it able to cause the disease in the plant. So this is these two talks on the systemic acquired resistance are just uh, you can say briefing on this big subject which on which you can find books or hundreds of paper and still people are not able to completely know how the systemic acquired resistance work, but lot of the things are known, and many of the things are applied to protect the plants against the pathogen. For example, by the use of NPR1 gene or by the increase of the expression of PR genes, the plants are protected against the pathogen. It is a vast subject, and a lot of work has been done, and there are many groups who are working on the different aspects of the systemic acquired resistance. So the purpose of these two talks are just to briefly uh, convey how the systemic acquired resistance work, but I will appreciate if you study more papers or the books on this topic, because this is one of the most important topic in the plant resistance. Thanks.